Once again, we will start with the next lecture of medicinal chemistry, that is lecture number 8, Parasympathomimetic Agent. Before that, we completed the sympathomimetic agents from the autonomic nervous system. So, let us start with the parasympathetic agents. So first of all, we should know what is meant by the parasympathomimetic agents. It is the part of autonomic nervous system that controls and regulates the internal organs. As I shown here, it is also the part of autonomic nervous system that controls and regulates the internal organs. So this autonomic nervous system, it is nothing but the also called as visceral or the involuntary nervous system, as it controls the all involuntary actions and helps to maintain the constancy of the internal environment. So it works through the nerves, ganglia, plexus, and it regulates functions which are not under the control of conscious mind. So again, the ANS divided into the two parts, as we seen in the last lecture, sympathetic nervous system and parasympathetic nervous system. As we completed the sympathetic nervous system, so moving towards the parasympathetic nervous system. So what it mean? Parasympathetic nervous system, it is also called as a cholinergic system. As we seen in case of sympathomimetic drugs, the sympathomimetic drugs are also called as adrenergic system or adrenergic drugs. So here the parasympathetic nervous system also called as cholinergic system. So this system is one where the acetylcholine is used as a neurotransmitter. So one of the important neurotransmitter from this category is acetylcholine, and which is released in the body. So this acetylcholine has the work like it works with the peripheral nervous system and central nervous system. So it is a very important neurotransmitter from this parasympathomimetic category because it has the function or it plays the role in both parasympathetic peripheral nervous system and in the central nervous system. Now moving towards the next slide, the cholinergic neurotransmitters. Cholinergic neurotransmitters, as I told, cholinergic neurotransmitters are nothing but the parasympathomimetic neurotransmitters. In that, one of the important is acetylcholine neurotransmitter. So it was first invented by the scientists Red Hunt and Thiewy in 1906, and it is the major neurotransmitter at postganglionic synapses of the cholinergic or parasympathomimetic nerve endings. As I shown here, this is the neuron, complete structure of neuron, and this base is as elaborated here, and these black dots, these represent the release of acetylcholine in the parasympathomimetic neuron. And here this blue represents the sodium and yellow dots, these represents the calcium release. So this is the position where acetylcholine is released in the neuron. Now moving towards the next, biosynthesis of acetylcholine. So the biosynthesis of acetylcholine is carried out by flow of this reaction. So as I shown here, first the serine, which is the amino acid. Serine nothing but amino acid. So serine is treated with the ethanolamine, or you should say 2-amino-ethanol. Serine treated with the 2-amino-methanol in the presence of enzyme serine decarboxylase. So this is a first step reaction which is carried out in the liver. So there is a formation of choline, which is a second step process, and this choline is obtained in the presence of N-methyl transferase. Further, choline is treated with the acetyl coenzyme A, and there will be the formation of final acetylcholine, which is the biosynthesis of acetylcholine. So this is a very simple reaction which is carried out only in the two steps. Moving toward the catabolism of acetylcholine. So first of all, we should know what is meant by the catabolism. So catabolism is nothing but it is the sequence of enzyme catalyzed reaction. So enzyme catalyzed reaction, which is denoted by the breakdown of the large molecules into the smaller one. So the large molecules are degraded or the broken down into the smaller one, that is the catabolism process. So here catabolism of ACH, that is the acetylcholine, can be carried out with the help of enzymatic hydrolysis. Means the various enzymes undergo the hydrolysis process, so the acetylcholine catabolism is carried out. Here the cholinesterase enzyme which rapidly hydrolyzes the acetylcholine into the choline and acetic acid. As I shown here, this acetylcholine in the presence of enzyme cholinesterase divides the acetylcholine into choline and acetic acid or acetate. This cholinesterase is also called as acetylcholine esterase. So this cholinesterase also known as the acetylcholine esterase. So it is a simple process where enzyme undergo the hydrolysis. So the acetylcholine is split or divided into the choline and acetate or acetic acid by the presence of cholinesterase enzyme. And this cholinesterase also called as a acetylcholine esterase. Further next point is that acetylcholine acts on the two different classes, or it consists of it will roll its function on the two receptors. First muscarinic receptor, second is the nicotinic receptor, or we may say the acetylcholine play its role on the two receptors, M receptors and N receptors. We will see what it means. So first is the muscarinic receptors. Muscarinic receptors belongs to the family gamma or G protein coupled receptor. In the sympathomimetic agent, when we seen the PPT and the lecture of sympathomimetic category, in that I have already shown you how the G protein coupled receptor work, where it's situated and its structure. So this it is also as the alpha and beta receptors belongs to the G protein coupled receptor. Similarly, muscarinic receptor belongs to the G protein coupled receptor. If you want to know regarding the detail of G protein coupled receptor, you should refer the lecture number 7 from the same class. So muscarinic receptors are more sensitive to the muscarine than the nicotine. As compared to the nicotine receptor, these are the more sensitive. These are nothing muscarinic receptors. These are the water soluble toxic derivatives and which are obtained from the mushroom category of Amanita muscaria. So these are obtained from the which source? Mushroom source, that is Amanita muscaria family, and which causes the activation of peripheral sympathetic nervous system. So important is that M receptor, the importance of this M receptor is that it acts on the peripheral sympathetic nervous system. Okay, so next there are the following 5 types of the muscarinic receptors as I shown here: M1, M2, M3, M4 and M5. We should study these 5 receptors comparatively in order to avoid any confusions, and we should study these 5 receptors in the 3 categories. First, if they are having any synonym or indicated by any other name. Second, by the location where they are situated. And third, the role which they will play. So we will see one by one. First one is the M1 receptor. So this M1 receptor also called as a neural receptor because as these receptors are in the neuron. So on the basis of that, these are also called as a neural receptors. M2 receptors having the another name, that is a cardiac muscarinic receptor. M3 also called as the glandular muscarinic receptors. Again their location, first M1 where it is located. This receptor in case of the parasympathomimetic agent or parasympathomimetic nervous system, these are located in the exocrine glands and autonomic ganglia. M2 receptors are located in the atria and the conducting tissues of the heart. So on the basis of the name cardiac, obviously they are located in the heart. M3 exocrine glands and the smooth muscles. M4 and M5 both are located in the central nervous system, but there is a difference that M5 specially located in the substantia nigra part of the CNS. Now moving towards the function which they will play. First, M1 receptor, it affects the arousal attentions and emotional responses and affective disorders. So these are the role played by M1. M2 obviously the cardiac related. M3 it having the function in the lacrimal salivary secretions and having the mostly stimulatory effects. And M4 they have the direct regulatory actions on the potassium and calcium ion channels. And the last M5 it regulates the dopamine release. So it regulates the dopamine release. So these are the 5 types of the M receptors or the muscarinic receptors with their location, function and other name. Moving towards the next second type of the receptors from the parasympathomimetic or parasympathetic nervous system, that is nicotinic receptors. As I told, muscarinic receptors are the more sensitive than the nicotinic receptors. Means we may say that the nicotine is less sensitive than the muscarinic receptors. Next one, that these receptors are derived from the nicotine. On the basis of the name, these are derived from the nicotine, and they does not stimulate the nicotine receptor but binds to the receptor. So there is a specific role regarding this receptor is that these receptors does not stimulate the nicotinic receptors but they binds only. Next, these receptors are the rosette like, as I shown in this figure, rosette like the rose flower and having the pentameric structure. Pentameric means only the 5 petals. So these are the rosette like pentameric structure which encloses a ligand gated calcium channels, calcium as well as the potassium channels. Here as I shown inside this, when this pentameric structure opens, it will releases potassium and calcium channels, ions. So this opening of this pentameric structure or the N receptor causes a rapid flow of the potassium and calcium ions, means the depolarization going to be happen there, and that results to the generation of action potential. So as I shown in this figure, this is the top view, this is the top view of the pentameric structure, this is a side view, this is a closed structure of the nicotinic receptor, and this is the open structure of the nicotinic receptors, and from this center there is a release of potassium and calcium ions leading to depolarization and leads to generation of action potential. So this is related with the nicotinic receptors. Now moving towards the next slide, that is the types of the nicotinic receptors and the role they play. So see, as we see in the muscarinic receptors are divided into the 5 types. See, these nicotinic receptors divided into the 2 types: muscle type nicotinic receptor, neuronal type nicotinic receptors, or we may say NM or NN. Moving towards their exactly the difference with their role. So as I shown here, I will expand this PPT. So first, muscarinic nicotinic receptor, muscle type nicotinic receptor and the Neuronal type nicotinic receptor. First we will focus on the muscle type. So on the basis of the name, these are the neuromuscular, exactly present in the neuromuscular junctions. It will cause the contraction of the skeletal muscles. Again, opening of the cations, calcium and potassium ions. They having the agonist role with the nicotine and antagonistic role with the next category. Now next, that is a neuronal nicotinic receptor or neuronal type nicotinic receptors. These are present in the autonomic ganglia. As muscle type, these are present in the neuromuscular junction, and this nicotinic neuronal type, these are present in the autonomic ganglia. Again, these are also present in the adrenal medulla and having the role of releasing catecholamine. They release the catecholamine. Again, they helps in the excitation of central nervous system as well as their inhibition. Again, same function, that these also helps in the opening of the potassium, calcium and sodium ions, and here only the potassium and sodium ions. And in this case, in case of nicotinic neuronal type, potassium, calcium and sodium ions. Again, these are also called as a DMPP nicotine, and last one, that the drug from this category, that is the hexamethone trimethinium. So this is some general important discussion regarding the nicotinic receptors, and with this we completed the overall parasympathomimetic nervous system with the introductory part. Parasympathomimetic nervous system with the introductory part. Moving towards the drugs and the classification from this category, we will see in the next lecture. So thank you everyone.